All right. All right, so this is a commentary to go along with your Unit 1 review questions. And so I thought I'd just go over the questions real quickly, give you an idea of what exactly we're looking for, so you can actually answer the questions and review the material. All right, so if we're looking at question number 1, what would be some positive and negative consequences of a mass extinction that suddenly eliminated all microorganisms? And so sort of the point of this question is get you to review a little bit in the first half of Chapter 1, where we talked about the many positive uses for microbes. Hopefully by the time you finish looking at the material and answer this question, you'll notice that there are really only 1 or 2 positive consequences of a mass extinction of microorganisms, and that the vast majority would be negative. So just make sure you go back over and talk about the different ways that microbes have affected the environment and the different ways that humans use microbes. All right, number 2. Describe the contributions of Redi and Pasteur to the defeat of the theory of spontaneous generation. So if you need to review this, you can take a look. In Chapter 1, there is an insight 1.2 on chapters 12 and 13. And on page 12, you'll see Redi's experiment where he had the rotting meat, and he had a lid on one dish and a covering missing from the other dish. And then he went on to observe where he saw maggots. So that was that experiment. And then Pasteur's swan neck flask experiment is explained to you on page 13. Remember that both of these were an attempt to disprove spontaneous generation. Spontaneous generation was the belief that simple organisms arose from non-living material. So the point of this question is to explain how these 2 experiments helped defeat that concept. Remember that the opposite of spontaneous generation is what we call biogenesis. And so biogenesis states that all forms of life arise from other forms of life. So, for instance, fly larvae come from the eggs of flies that have had intercourse. However, well, they don't technically have intercourse, but you know what I'm saying. That the baby flies, the larvae, arose from a male and a female fly. All right. Moving on to question 3. In the field of taxonomy, how are the names of organisms derived? And so this starts off with our taxonomy here. And if you take a look beginning on page 17 and growing through pages 21 and 22, you'll see an introduction to taxonomy. If you take a look on page 28, you'll see how we assign specific names to microorganisms. So what we practice is something called the binomial system of nomenclature, or just binomial nomenclature, giving each living organism 2 names. The first name is the genus name, and the second name is the species name. All right. 4. When discussing some prokaryotes, what is meant by the term extremophile? So you remember the term extremophile just applies to the fact that there are certain microbes that can live in conditions that are so extreme you probably wouldn't think anything would be living there. And hopefully you picked up in the lectures or the outlines that we often use the term extremophile to refer to the archaea. And we mentioned 3 types of archaea: the methanogens, the halophiles, and the thermophiles. So make sure you review that and make sure you know where you might find these organisms living. All right. Then we've got question number 5. That's again about some prokaryotes. Describe the feature of chlamydias and rickettsias that makes them unique among the bacteria. And then what other group of microorganisms possesses this characteristic? Well, if you go back and you look at the chlamydias and rickettsias, those are bacteria, so they're in chapter 4, you'll notice that they were referred to as being exceptional because they were obligate intracellular parasites. And the second part of the question is asking you what other group of organisms did we talk about in this unit that were all obligate intracellular parasites. So this is to compare two groups, the viruses and then these two couple of bacteria. So make sure you review that. Question 6, compare and contrast the bacterial gram-positive cell wall and the gram-negative cell wall. Okay, so this is back in our prokaryotic chapter, chapter 4. And on page, the figure I like is on page 97, figure 4.14, where you are comparing the structure of the gram-positive and the gram-negative cell walls. All right. Also, why is the gram reaction of importance in diagnosis and treatment of an infection? We talked about several things here. We said that first off, performing the gram stain is going to allow you to eliminate a great deal of possible suspects that might be causing a bacterial infection. We also said it was important because some antibiotics, if you remember, aren't necessarily going to be able to cross the gram-positive or the gram-negative cell wall. It just depends. And we also mentioned that there was a toxin on the surface of the gram-negative outer membrane called the LPS, or lipopolysaccharide. And that's of importance because it's a dangerous toxin to humans, and all gram-negatives possess this toxin. All right, number 7, differentiate between mold and yeast. So what we're talking about here is the single-celled yeast versus the mold with their hyphae and their mycelia. So you want to differentiate that way. You also might want to mention that the yeasts are capable of budding, which the other fungi do not reproduce that way. Why are many fungal pathogens described as dimorphic? The term dimorphic just means that the particular fungus exists in two forms, meaning under one set of conditions it is a mold, and under another set of conditions it is a yeast. And a good example of a fungal pathogen that is dimorphic is going to be Cryptococcus neoformans, which we spoke of. Remember, it's a basidiomycota, and it exists as a mold in the soil, and you breathe in the mold spores into the lungs, and then the mold spores sort of shift and start to appear like yeast and start to grow in the human body as yeast. So that would be an example of a dimorphic fungus. Right. Number 8, imagine you could design cells for research that are missing one particular type of organelle. For each of the following, predict in detail what would occur in a cell missing that organelle. So the point of this question is just for you to go back at the beginning of chapter 5 and just make sure you know the roles of all of the organelles. And I was just trying to be a little bit creative. What would you think a cell would be like if it didn't have ribosomes? What would a cell be like if it didn't have a nucleus? So this is just to help you practice the structures of the eukaryotic cell, making sure you know them. Also, remember I added a web tutorial comparing prokaryotic cell structure and eukaryotic cell structure, so you might want to take a look at that. All right, number 9. For each of the following general groups of microorganisms, construct a definition based on features that allow you to tell the difference between them. So there will be a section where I'll make statements, and you have to tell me whether that statement applies, say, to a fungus, or does that statement apply to a helminth, or does it apply to a whole group of organisms? It could apply to the protozoa, the algae, and the fungi. So you'll have multiple answers that you can give, but the point of this question is that you need to go back and review the general structures. For example, if I gave you the statement, these organisms possess a cell wall composed of chitin, then you would be able to tell me immediately that that was the fungi. However, what if I said these microorganisms are eukaryotes? Well, then your possible correct answers are fungi, helminth, algae, and protozoa. So you'll have a few statements about these various groups based on what we've learned about their general structure and their general biology. And so you'll need to be able to differentiate between them. All right, number 10. With detailed examples, explain how the ability of a protozoan to encyst affects the pattern by which the pathogen is transmitted. So in this question, we're wanting you to indicate, for example, if I give you a protozoan that forms a cyst, you need to be able to tell me how it can be transmitted from one person to another. So remember, if a protozoan could form a cyst, that means that the organism can be transmitted indirectly, meaning that the organism can live in the environment in its cyst form and eventually be picked up by another host. But your two hosts had no direct contact, so that was indirect transmission. And example of a non-cyst former, which is Trichomonas vaginalis, since it doesn't form a cyst, it has to be directly transmitted. So what we're saying is it requires direct mucous membrane to mucous membrane contact between the two hosts. So make sure you review what a cyst is and then take a look at that. All right, 11. Describe how the anatomy of a tapeworm is indicative of this organism's lifestyle. So remember, we said the lifestyle for the tapeworms is one of feeding and breeding. And when you go back and review the structures of your tapeworms, you're going to be able to see that all of the organs and structures that you find are involved in both of those, feeding and breeding, reproduction. So that's all it's trying to get you to do, is take a look at the anatomy and to see how the anatomy fits in with the overall lifestyle of the tapeworm. All right, 12. Construct an argument supporting the classification of a virus as a non-living infectious particle. So to start out, you'll probably want to look at page 151 in your text, table 6.1, properties of viruses. You also will want to perhaps flip back to chapter 2, where they talk about the definition of life, and you'll notice that viruses do not fulfill any of those definitions. So we said viruses have no metabolism, they don't acquire materials and energy, they can't reproduce on their own, they lack a mechanism for making proteins, they're acellular, list goes on and on. They contain either DNA or RNA, but never both. All of those things make viruses very, very different than our living organisms. All right, 13, what are the implications of latency in terms of the spread of a viral pathogen? And what we're shooting at here is the fact that we know that some viruses can insert their DNA into the host DNA, or we use the term integrate commonly. And we know that once the DNA of the virus has integrated, then every time that cell divides, it will replicate the viral DNA as well. So any of the progeny of that originally infected cell, any of those progeny are going to have the viral DNA and therefore can be activated, and ultimately the process of viral replication can occur. So even though you don't have viruses actively replicating and infecting cells, you are passing down the viral DNA from one cell to another. All right, and then number 14, in detail explain how fungi, protozoa, and how bacteria, fungi, protozoa, and viruses are arranged into taxonomic categories. And you should probably have helmets there as well, because that's something that we studied. So again, you need to make lists of the divisions and classes that we find when we're looking at the organization of these of all of these organisms into groups. So remember, when we were looking at the bacteria, we were mainly looking at the type of cell wall, if it's present at all, when we were placing bacteria into groups. With the fungi, it was the production of sexual spores. For the protozoa, remember, it was motility. And with the viruses, remember, we were looking at three major things. Is the virus an RNA or DNA virus? Is the virus enveloped? And then what is the nature of the nucleic acid? Is it double-stranded? Is it single-stranded? So make sure you go through those taxonomic categories, and you can also identify the examples that I gave you that go along with the various divisions and with the various classes, which those would be the helmets. All right, so that's what you should be aiming at when you're going over these questions. They're not necessarily a simple question to answer because some of them are quite involved, but that's to help guide you as to the material you should study for your test. All right, guys. I'll see you next week.